Afternoon, good evening, and good morning, everyone, wherever you might be. I am Rob Burgess, and I'm Chief Business Officer for Sino-Biological. And it's my pleasure, and I'm delighted to welcome you to the next installment in the Sino-Biological webinar series titled Lock and Key Immunodetection Technologies. We have an excellent speaker today whom I'm going to introduce in just a minute to talk about some very cutting edge technologies from a new company. Before I do that though, I just want to address one housekeeping issue, and that is we ask everyone, if you have a question, you are certainly welcome to ask that question. Just please enter your question into the chat box. And at the close of the webinar, I will ask those questions verbally of our speaker. We found that that tends to be the most efficient method for addressing everyone's questions. So with that, and without further ado, it's my pleasure to introduce to everyone Dr. Heinrich Jensen, the founder and chief scientific officer of Fidabio. Heinrich is the founder, obviously, and CSO of Fida Biosystems, which is based in Copenhagen. And he has an extensive academic scientific research career, obtaining his doctorate from Aarhus University and completing his postdoc at the Swiss Federal Institute of Technology. He is also an associate professor at Copenhagen University. And he's published over 110 peer-reviewed publications and has received over 3,500 citations related to those publications. So the title of Dr. Jensen's webinar today is The Ultimate Biophysics Tool, Fast and Accurate In-Solution Analysis of Proteins and Nanoparticles. So Dr. Jensen, welcome. It's an honor to have you here. And I will now stop sharing my screen so that you can share yours.
Thank you very much, Rob. I will just start sharing my screen. Do you see my presentation now? Great. Just seeing if I can get my pointer to work. Sorry. Yes, so thanks, Rob, and thank you very much for inviting me to do this webinar. So Fida is about characterizing biomolecular behavior by monitoring changes in hydrodynamic ratios, and we can do so under native conditions. So this is the context of my talk today. So I'm going to give you some of the technology characteristics, and then, of course, importantly, tell you something about the application landscape, because we have a very extensive set of applications that we can do with this technology. Of course, I will explain the principle behind, and also briefly some application and the instrumentation that we are currently marketing. Just before we start, you see a graph here where it says hydrodynamic radius. So this is actually an important point of the presentation here, that we are measuring, we have an absolute readout. We are measuring a size of proteins and molecules and particles in solution. Typically, we measure it in response to something, for example, a titration, which will give us a lot of information on in-solution structure and interaction constants. So up here in the top of the slide, you see a picture of the instrument. So it's a big black box here, which is an autosampler. So we can measure, the sample format could be 96 well plate, it could also be vials, for example. On the side here, you see a detector box, and we can actually detect colored fluorescence. We can also detect UV fluorescence coming from tryptophans and tyrosines in proteins, for example. The actual analysis takes place in the upper compartment here. So the analysis takes place in a capillary, fused silica capillary, which is a consumable accompanying the instrument. And then, of course, we have software, dedicated software, where you can auto-analyze your data and also control the instrumentation. Most importantly, the Fida technology is gives you an absolute size determination in nanometers. So it's not a relative readout, it's an absolute size that you're measuring. It doesn't require any calibration. Also, the second important point here is that it's what we call a first principle technology. And by that we mean that if we know the system, we can actually exactly calculate the response. We can simulate how the raw data will look. And that's important because it will allow us to do very fast and efficient assay optimization, also on unknown system. As I mentioned, we can work with both label and label-free, so UV fluorescence. We can measure in any sample matrix. So the technology is completely invariant to the nature of the sample matrix. So we can, for example, measure in serum plasma, cell lysate, fermentation media, and so on. Because it's essentially a microfluidic system, we can work with small sample volumes from nanoliters to microliters, depending on the assay type. We are very sensitive in detecting size and size changes, actually down to only 5% changes in radius. And we cover a size range coming from small molecules, 0.5 nanometers, to large nanoparticles. So it basically includes small molecules, it includes all proteins, virus particles, liposomes, exosomes, and so on. In terms of affinities, we cover the range from picomolar to millimolar. I already spoke about the autosampler. One important point is that we have temperature control, both in the sample tray, but also in the capillary chamber. So for example, we can do thermodynamics. We can also store the samples at low temperature and do the analysis at a higher temperature. For example, store them at 5 and measure at 25 or 37 degrees. This is what we call the application landscape. And of course, we can do simple binding measurements, multiple binding, what we call non-binding system, which could be conformational assessments, particulate work like AAVs, nanodisk, liposomes. Recently, we also started looking at biological condensate, so liquid-liquid phase separations. And then we have the others here, which I will go into a bit more detail with today. So, for example, clone selections, where we can do clone selection, where we select both the clone in terms of its biophysical properties, its ability to bind the target, but also the ability of the clone to actually produce the protein. The other point I will go into more details with today is membrane protein. We have a recent application here where we do detergent screenings on membrane proteins to find out which detergents are most efficient for solubilizing your membrane protein. And then the final thing I'll go into more detail with is multi-specific antibodies and targeted protein degradation, which are both examples of multiple binding systems where we form ternary complexes, and it's something that we have integrated recently in the software package. And of course, if you're interested in any of the technologies, any of the applications that I will not talk about today, then please do drop me an email, and I'll be happy to go into a dialogue with you on all the other applications that we won't have time to cover today. So essentially, the Fida technology is based on a hydrodynamic flow in a few silica capillaries. So these capillaries standard capillaries are 75 micrometers in diameter. When we inject a small band of samples, so in the order of 40 nanoliters, and subject it to a pressure-driven flow, it will adopt this parabolic flow profile. And that means that we have a diffusion gradient in the front and in the back here. So molecules will diffuse here. If they diffuse fast, then this dispersion will be evened out, and we measure a narrow peak signal here when we measure the fluorescence by the end of the capillary. On the other hand, if it diffuses slowly, it will tend to stay at the edge here and in the middle, and we observe a broad signal shape here. Now, if something binds to the protein down here, it will change signal shape and move up here. And this is essentially how we are probing binding interactions. Let's say that we are measuring in plasma and serum. That will give us a lot of residual fluorescence. But since we are actually looking at a signal on top of the background and only looking at the signal shape, that means that we can measure in almost any sample matrix and get the same results or get the same performance as we get in simple buffer systems. So this is an example on how the raw signal will look when we are measuring a binding interactions. If there's no binding, in this case, we're looking at fluorescein binding albumin. We see a sharp signal shape. And then as we increase albumin concentration, we observe a much broader signal shape here. And when we analyze this signal shape using the algorithm in the software, we can extract the first the diffusion coefficient, which we use to calculate the hydrodynamic radius. And that gives us this binding isotherm here. When we fit the binding isotherm in our software, it gives us information on the size of the binder, the size of the complex, as well as the interaction constant. We also get some other additional readouts, such as the area under the curve here. And this gives us a lot of QC parameters of the technology. And sometimes it also gives us an orthogonal readout of, for example, a binding, if the quenching of the fluorophore changes upon binding. So one of the important things of the Fida technology is that it offers us a lot of ways to QC our assay. First, of course, as I already mentioned, it's the fact that we're measuring absolute size that gives us a reference point, and it tells us what it is that we're measuring. Peak area tells us about sample loss. We can accurately count the number of aggregates in our sample, and we do this every time we do a measurement. So every measurement is associated with an aggregation count. If we are working with labeled material, we also get a labeling quality. We can see if we have difficult proteins that sticks to surfaces. That's also visible from the raw data. We measure viscosity. This is important, for example, in weak interactions, where we need to compensate the effect that the binding partner has on the viscosity of the sample. We measure several species, several size distribution, and we can also provide a polydispersity index. And then finally, as I will talk about later, we have a PDB correlator, which actually links up with the protein data bank and allows us directly to link the measured hydrodynamic radius to a structure of the protein or the protein complex that we're studying. In terms of application, I will start out with a simple application here. It's insulin that we put a label on. So it's fluorescently labeled insulin, interaction with an antibody against insulin. In the absence of the antibody, we measure a size of 1.4 nanometers. That tells us it's monomeric insulin. Insulin can also exist as a dimer or even a hexamer, but this is monomer because it's 1.4 nanometers. As we titrate along, increase the amount of the antibody, we end up here at a plateau around 5 nanometers. So this is the complex between insulin and anti-insulin. You can also take a mixture up here and titrate further with a secondary antibody. And that gives us then a new plateau at a larger size because it's two antibodies and insulin. So here we're around 8.5 nanometers. So it's just to illustrate that we have this technique where we can build large protein constructs in solution and then quantify the individual events, binding events leading to the construction of these in solution constructs. Another example here is where we look at conformational changes. This is maltose binding protein that is labeled. It's titrated with maltose, a small molecule. When MPP binds maltoses, it changes conformation. It actually becomes smaller, as we can see from the curve here. So even though the mass increases, the complex becomes smaller due to conformational changes. So it allows us to characterize the complex in terms of size and also in terms of KD. The size change here is only in the order of 10%, but that is more than enough to characterize the binding. Another example here is a recent example from a collaboration we have with the National Physics Laboratory in London, Professor Morais, where we look at membrane detergent screening. So here we take directly a cell lysate containing membrane protein, and we use different detergents to solubilize them. So this is a DFP tagged membrane protein, and from the area of the solubilized protein here, we can see how much goes into solution. We can also determine the size and the polydispersity index. And particularly in this collaboration, the polydispersity index was important because it could tell us something about the ability of the membrane protein to crystallize. So the important thing here is that we are doing it directly in cell lysate without the need for purification. And in this campaign, we screened 12 detergents. And because of the microfluidic nature, we only used 1.5 microliters. And for 12 detergents, we did a full scan in less than 3 hours. We have the option of cooling the samples while we're doing the analysis at 5 degrees, and then only spending a few minutes of doing the actual size estimates at 25 or 37 degrees. This was a DFP tagged membrane protein. We can also work with his-tagged membrane proteins. In these cases, we use a non-covalent his-tagged dye to quantify the membrane protein and also to look at the size of the membrane protein. A second application here is clone screening. And the difference here compared to other technologies is that we determine both the titer and the KD, so the interaction constant with the target, in a single assay without any purification. So basically what we do is that we spike in the fluorescent labeled targets in cell supernatant and measure the size. And from the size, we can tell the degree of binding. So if it's a full complex size, it tells us that the membrane protein is fully bound, and that could be due to either a very tight binding nanobody that we screened for in this case, or a very high expression level. The second step is then to further characterize these candidates in a competitive titration format. And this competitive titration, where we titrate in non-labeled targets, gives us both the KD and the expression level. And the analysis of these curves is completely incorporated in the software. And based on these results, it's possible to select the most efficient clone and use and purify that and possibly do further analysis and characterization. The final example here is actually for ternary complex formation, and that's relevant, for example, to bispecific antibodies. It could also be relevant for targeted protein degradation. So in this case here, we are looking at bispecific antibodies, and we tend to label the antigen in this case here. And when we do a titration with the bispecific antibody here, we, of course, at low concentration just measure the free antigen. There's an optimum concentration where we measure primarily the ternary complex, and then a high concentration where we measure primarily the binary complex. These data can be fitted directly in the Fidabio software, and from a single experiment, we can get a lot of information on size of the different complexes. We can measure all the affinity constants corresponding to the formation of these complexes, and importantly also the cooperativity. A related example here is targeted protein degradation. This was done in collaboration with C4 Therapeutics, a Boston-based company. And also here we fluorescently label the protein of interest and keep a constant concentration of the E3 ligase and titrate with a concentration of a small molecule linker between these two proteins. And again, we see this bell-shaped binding curve that can be analyzed directly with this software module that I have given you a screenshot of here in the presentation. A few words about the actual instruments. So as I already mentioned, we can work with vials, but also with 96-well plates. These trays are temperature controlled, and also the capillary chamber, and it's independent temperature controlled. The assay design is composed of 3 parts: the assay design, data analysis, and binding curve. Assay design allows us to calculate an experiment, and that's because of this first principle nature of the technology. And then, of course, we have data analysis. This is an example from a data analysis where you can see we can see up to 3 different size distribution. In this case, that's 2. We also see here these spikes here. Each spike here actually corresponds to a protein aggregate. So every time we do a measurement, we calculate also the number of aggregates in the sample. So that's a QC check of the sample, and sometimes the aggregation itself could also be a purpose of the investigation. And finally, there is this protein data bank correlator. It's a tool in the software where we can just enter the PDB code if that exists of your protein or complex. The software will then give you a predicted RH value. You can also load your own PDB values, for example, from Pymol or other kinds of software, calculated structures. And that way use those to obtain a predicted hydrodynamic radius that can then be subsequently be compared to experimentally measured values. This was what I had chosen to present for you here today, and would be very happy to discuss further any of your questions.
Thank you, Dr. Jensen. That was a wonderful presentation. Very exciting technology you have there. And
thank you.
Company and Fidabio. We've got several good questions from our audience members, and we have a wonderful turnout today. So I'm just going to start with a scientific question. And while we're asking this question, I do encourage everyone else who is still attending to type your questions into the chat box, and I will get to them verbally one by one for our speaker. So the first question comes from Grace Lee. It's a great question. It's about your dissociation constant slide. And Grace is asking, how long does each assay take to get a full dose response and curve to measure KD?
Yeah, that's a good question. So it depends. It depends a little bit on the system, but let's say it's proteins below about 20 nanometers. Then each measurement points take in the order of 3 to 4 minutes.
Okay. Very quick.
And then it of course depends on how many points you want. If we are looking at binding to larger particles, like for example an AAV particle, the measurement will take longer, and that's because larger species like nanoparticles, they need more time to diffuse. So we are probing diffusion. So very large particle will take longer in the order of 10-15 minutes per sample.
Still very quick, rapid assay. Great. Next question is from Ross Walton. Ross asks, what's the advantage of your sizing method over DLS?
Yeah, that's also a great question. So I think there are several advantages. So one of them is that we are using very small amount of sample. So for example, we can size in as little as 20 nanoliters of sample. So if you have very small amount of your protein or your AAV and so on, we can still work with that and get very, very nice results. We can look it up to 3 size distribution, and we can determine polydispersity index. That's quite similar to DLS. So I'll say that the main point is that we can measure in very low sample amount, and then of course also we can measure in complicated sample matrices. So we can, for example, measure your protein also in serum and plasma.
Yeah, so the type of sample is not any kind of rate limiting factor, I guess.
Yes, exactly. And then of course you could add also the automation aspect. So we can run up to 192 samples unattended. You may be able to do that with some DLS equipment also though.
Excellent. Next question is from Christine Bain-Wind. And Christine asks, can this technology identify different forms of a protein, such as monomers, oligomers, and aggregates?
Yes, so I think concerning the aggregates, we actually had this is actually an example of that. So if your protein aggregates, we will give you an exact count on the number of protein aggregates in your sample, and we have multiple applications on this. We also have application where we look at monomer, dimer, hexamer equilibrium. We have done this for insulin. We have done it for different protein system also. So yes, and it will be able to provide you with also the interaction constants that are associated with dimerization and so on. And you can link the structures if you have structures of your oligomers, you can also link the structure to the in solution measured hydrodynamic radius.
Mm-hmm. Next question is from Eric Lee. Eric asks, is the KD measurement comparable to that of Biocore?
Yes, yes, it is for many systems, particularly if you have simple one-to-one interactions. Then we measure very comparable results, and we have confirmed that in multiple studies. We have also seen in some studies, particularly if you have multiple binding associated with your protein, then we may see differences because the avidity effects will affect the results in different ways, whether you look at it in solutions or on a surface. For example, if you coat a surface with an antibody, you have a very high density of binding sites, which could give you an apparently much higher association constants than you would see in solution.
And Halcano asks, can the technique substitute, hold on just a second, somebody else was asking another question. Can the technique substitute other usual procedures such as drug dosing in a parasite culture?
Yeah, I mean that's a very good that's a very good question. Also, I think we can bridge in vivo studies and more very classical in vitro biophysical studies better than many other techniques, because we can measure in very complicated sample matrix, so under native environments, and we can also measure binding to membrane associated proteins, to particles and so on. Whether you can actually substitute it, I think that would require some more experiments to firmly answer that question.
Josinato Illas asks a question. I think this is about the potentially disruptive aspect of your technology, and that is, can the Fida technology actually replace a traditional ELISA? Do you think that's possible?
For some applications, I think this is for sure possible. So again, one of the things one of the advantages is that we can see the binding using just one ligand. So we have, for example, an application, I did not discuss it here today, but we have an application where we're looking at auto antibodies in plasma serum. We have application where we are looking at drug monitoring of antibody-based drugs in plasma serum from patient samples. These are all published results, so if you're interested in learning more, I'll be happy to send you the publications, and also to discuss this aspect further.
Great, we can put you in touch with them if need be. Grace Lee has another good question, I believe. How low can the molecular weight of the small molecule go to detect a change in RH of the protein of interest?
Yeah, I mean, it depends a lot on the actual system, but this was one example. So I think I forgot to say it was made with one of our customers in Switzerland, the Fachhochschule. So maltose binding protein is 42.5 kilodalton. So this is where we put the fluorescent label, and maltose is a small sugar. It's 360 dalton, and here we see a very clear change in size, and that is because we also have a change in conformation when maltose binding protein binds maltose. In some cases, we have very little or no size change, and in those cases we tend to label with an environmental sensitive dye, and there we use instead the fluorescence intensity, which is also visible in this example here, as a probe for looking at the binding to small molecules. So the molar ratio you see we calculated here is the molar weight ratio is 118 to 1 in this particular case.
So this is more of a business model related question from Halcano, and I think it's about distribution and placement of the equipment, which means, do you place your equipment maybe in common cores, or do you only just allow like a direct purchase? What's your model for getting the system out into the world for use?
Oh, so I see on that point, we are very flexible because we really would like as many people as possible to use the technology. So I mean, we are focusing in Europe and the United States. At the moment, we have a sales force in Europe. We have sales force in the United States and a growing group of field application specialists. We do in-house demo where people send the sample to us, and we also do remote demos where people will have an instrument for a period of time to test it out on their application. And of course, we also invite people to visit us in Copenhagen, and we very much like to have visitors at our facility to have a go with the instruments and also see how the instrument is made and how we use the software and so on. So we're very open to that. So if you're interested then in trying the instrument, then please do get in touch with us.
Yeah, good excuse to visit Copenhagen for all of our attendees, right? So Eric Lee stole my question actually, and it was more of a business model question, and that is like, what's the list price of the unit?
Yeah, so the list price. I mean, we have different versions of the list of the instrument, but just to give you a ballpark numbers in euro, is around 135,000.
Okay, good.
So I guess it's slightly more in US dollars.
Yeah, I can't remember the conversion rate, but
they are not far, the euro and the US dollars at the moment, I think.
And that's just a general cost. I mean, you have multiple versions of it, but that's your average list price, I guess.
Yeah, that's a good estimate, I think. Yeah.
Great. Christine has another good question here. Has your technology been used in the context of clinical development, such as the monitoring of pharmacokinetics or pharmacodynamics of a drug?
Yeah, that's actually a very good question. So we have done I alluded a little bit to it before. We have done a model study, kind of a retrospective study on a group of patients receiving adalimumab. So this is an antibody-based drug, where we were able to actually measure the amount of adalimumab in the blood of these patients, but also at the same time measure the level of autoantibodies. So both neutralizing autoantibodies and also binding autoantibodies. So it was not an actual clinical trial, but it was real patient samples that was used to illustrate that the technology could be used for that. It is an area that we are looking more into. We don't have that many applications in this area yet, though.
So from a little bit of a selfish perspective at Sinobiological, we've always struggled with the manufacture of certain membrane-based proteins. And I was just curious, we were fascinated by your efforts to identify detergents that solubilize proteins that tend to form inclusion bodies or aggregates and so forth. So I just have to ask, did you identify like a secret detergent that works well on most proteins, or can you elaborate a little bit on your results from that?
No, I can't say we did that. And I mean, to be honest, we're developing the technology. So the expert knowledge on the membrane proteins, that's with guys like you, Rob, and of course with in this example here, we worked with Moraes and Johnson from the National Physics Laboratory in London. And the study we did here was with a known system. And the detergent we identified in this particular study were the same that were identified as the best detergent using other technologies like FSEC, for example.
Well, we'd love to work with you on that. Give us a chance to collaborate, right? We can pick some tough membrane proteins and see what magic.
We offer the challenge.
Somebody asked, is Fidabio and Sinobio the same company? And I have to clarify, no, we're not, but we're probably going to work together in that respect. So
it'll be a pleasure.
Looking forward to that.
Yeah.
I just want to hit home the major points and advantages of your instrumentation platform and technology over that of your competition. Can you tell us like the main one or two or maybe even 3 points that really separates your technology from that of the competition?
Yeah, I mean, you probably get slightly different answers depending on exactly who you're asking our company. But for me, the important thing is that it's an absolute technology that is a first principle technology. So we have a direct link to the theory behind the technology. We can calculate the response of our experiments. So that means we have 100% control of what it is that we're measuring. So I think that's the core of the technology, and that has some associated benefits. The fact that it's a microfluidic system means that we can work with very small sample matrices. We can work in complicated sample matrices, so we can do screening directly in cell lysates. We can measure in plasma serum. Sometimes your proteins doesn't behave the same way in a phosphate buffer as it does in serum, and these are questions that we can answer very rapidly. So we don't need extra development to transfer the assays to other sample matrices.
Sounds great. Well, on that note, I think we'll wrap things up just a little bit early today. First, I want to thank all of the attendees for joining us. We really had a worldwide crowd here today. I think it makes it more fun if I just call out the countries that attended. Of course, the United States, Germany, Sudan, folks from Cameroon, Kenya, and Mali. So we appreciate the worldwide presence and also the excellent questions from our attendees. Very much appreciated. Dr. Jensen, we want to thank you for giving this webinar today and telling us about your very exciting technology and the success of Fidabio. Congrats to you on your success in academia and industry. It's very inspiring. And
I have to say thank you very much to you also. It was really a pleasure presenting here today, and also thank you for the questions. And please get back with us in case there are further questions or essays that you would like to discuss in further detail.
Yeah, we definitely will. We'll put you in touch with any of those that we didn't get to today, which I think we covered most of them. Just to remind everybody, the recording of this will be available very shortly on the Sinobiological website. You just have to go into the webinar section there. And then finally, I just want to thank my colleague, Christine Lay, and our marketing department for organizing and executing this webinar. Great job today, as always, Christine. We appreciate it. And with that, I think we'll say goodbye and good evening or good morning to everyone, wherever you might be. So thank you very much.